All right, so now we are in chapter 1.3, and we're going to take a look at the specific types of microorganisms and talk about some of their specific characteristics and things that make these different groups unique. So first of all, in this portion of the chapter, they talk about the different sizes of organisms. So most of our microorganisms are unicellular, but some are multicellular, and we've already spoken a little bit about that, and we're going to look at that in more detail in just a moment. But remember what we said at the very beginning of the chapter is that microorganisms, by their name, are very, very small. So most of them we can't see without a microscope. In the text, in the OpenStax text, there is a nice image there, and on the image it shows the logarithmic scale. So everything from 0.1 nanometers all the way up to a millimeter. And it has different types of cells within that kind of table there. And so you can see that at our 0.1 nanometers, we have an atom. And then going all the way up to our 1 millimeters is where we see our frog egg, for example. And just before that, we have our human egg and pollen and an animal cell and plant cell. And basically the idea here is that we can see that there are different sizes of cells. So everything from an animal cell, which is relatively large on this kind of scale, again, microorganisms, so still can't be seen without a microscope, however, a little bit larger than an atom or a molecule or, say, a protein, for example, or a virus. So in order for it to be visible to the eye without a microscope, it needs to be at about 100 micrometers. So this is what allows it to be visible to the eye. And so we don't see a lot of our microorganisms that are visible to the eye. So many of them cannot be seen without a microscope. But we do see things, I said at the 0.1 nanometers, according to the text, you can see it in the table there. I'm not going to reference a page number because they change somewhat relatively often just based on the addition. So in chapter 1.3 here, we can see on the table the 0.1 nanometers is where we see the atom, which is quite small. This is nanometers. And then all the way on the other end of that table, we have our 1 millimeters, which is relatively large. And that's where we can see a frog egg as the example in the text. And then just down here, we have our animal cell, our plant cell as well. And then down here we can see our viruses, various sizes. Viruses are very, very different in their sizes. Over here we have mitochondria, and then even further down here we have proteins, somewhat up here. So you can really see in the text there kind of the different sizes. And again, at 100 micrometers is when we can really start to actually see these things. So with a visible eye, like a human egg or a frog egg, for example. So there are some structural differences within different microbes. So our structural differences. So as we just said, there are different sizes, for example. So different sizes, anything from unicellular to multicellular. So some unicellulars all the way up here are larger, like frog eggs, versus something that's multicellular that would be all the way down near the atom, like a protein or a molecule. So different sizes, whether it's unicellular or multicellular. Of course, a protein isn't unicellular, it's much smaller than a single cell, but my point being that there are things that are unicellular that are large or small, and things that are multicellular that are large or small. So then also we have the differences between membrane-bound nuclei. Also the habitat, so we see differences in habitats between these different organisms. We can see differences in their metabolism. Metabolism is the chemical processes within an organism. So oftentimes when we're talking about metabolism, we're talking about the building up or the breaking down of things. And typically what we're talking about when we talk about metabolism is what they utilize for energy. Also different structures. For example, a virus is made up of proteins and RNA or DNA versus a eukaryotic organism or a plant or an animal that's made up of lots of different structures. And then different things like whether it's unicellular multicellular, or acellular. And we'll talk about these as we move forward into the different types of organisms. And then we find our microorganisms in all three domains. So all three domains. So throughout the semester, we're going to be taking a look at all three domains. What are those three domains again? Do you remember? Bacteria, Archaea, and Eukarya, right? So we can see in both Archaea, bacteria, and Eukarya that we have microorganisms in all three, of varying sizes as well. So let's take a look at all of these different organisms. Let's kind of classify them and see where they fit in the general scheme of things. So remember we said that they're prokaryotic microorganisms and eukaryotic microorganisms. So we're going to use those as our two kind of main categories at the moment. So we'll start with our prokaryotic. So remember our prokaryotic microorganisms. Remember, prokaryotic means that there is no membrane-bound nucleus. No membrane-bound nucleus. And when we're talking about our prokaryotic microorganisms, we're talking about unicellular organisms. So then what we can do is we can separate our prokaryotic organisms into our different groups here. So we can further classify them. We can get even into more specific groups, namely bacteria. And what's the other one? Good, Archaea. So let's start talking about bacteria first. So if we kind of zoom into bacteria here, remember bacteria, of course, because it is prokaryotic, has no nucleus, no membrane-bound nucleus, and it is unicellular, or they are unicellular, as we already know, because we're talking about prokaryotic organisms to begin with, so that's already given. So some things that are characteristic specifically of bacteria is that their cell walls, first of all, they have a cell wall. Second of all, it's made of peptidoglycan. So cell walls made of peptidoglycan. And we're going to get into the specifics of what exactly that means. There are particular proteins that are arranged in a particular arrangement. And so we'll take a look at those kind of linkages between the proteins and what that means with the peptidoglycan and other things that we might find. So the bacteria, their cell walls are made of peptidoglycan. Bacteria are found nearly everywhere. So everything from glaciers in the Arctic snow to the Arctic ice, all the way down to the heat vents in the ocean or the ocean floor, all the way into our sulfur ponds in Yellowstone. So we find bacteria all over the place, including on us and in us. So there are actually more bacteria cells on us and in us than we actually have human cells. So we find bacteria all over the place in very great numbers. So they're found nearly everywhere. However, only some of them are pathogenic. So, however, some are pathogenic. So what does pathogenic mean? So a pathogen, pathogenic or a pathogen, is something that causes disease. So something that causes disease. So a pathogen is something that causes disease, or pathogenic. Something can be pathogenic. So some bacteria are pathogenic, some bacteria are not harmful, and some are helpful. So then also we have some are photosynthetic and some are not photosynthetic. So we do have some bacteria, like cyanobacteria, that are photosynthetic, can just get their energy from the sun, from the photons from the sun, and some that are non-photosynthetic, that require different things like carbon sources, different carbon sources to get their energy and their carbon. So we have varying things here with bacteria. Now, I do want to go into a little bit more detail of the bacteria here for just a moment because we're going to be talking a lot about bacteria throughout the semester. So what I'd like to take a look at are the different shapes and the different arrangements. So first, let's take a look at the common shapes of bacteria. So when we talk about our common shapes, we find them in different shapes, and that's one of the ways that we can classify them, right? So if we took a look at the Bergey's manuals or in a classroom or a laboratory, the first thing we might do is we might put some on a microscope slide and take a look at it through a regular bright field microscope, which is what we would find in a typical laboratory or a classroom. And we can identify or start to identify bacteria based on these different shapes that we find. So some common shapes, the first one is spherical. And if it is spherical, so if it's a round shape, this is called a coccus. So when we are going through and we're talking about different bacteria, we might say Streptococcus mutans. So again, if we say something like Streptococcus mutans, we're saying the genus, which would be Streptococcus via our binomial nomenclature. And then mutans would be the species. When we say Streptococcus, notice in that genus we actually say coccus, and that gives us a clue as to what that particular bacteria would look like. So if it says Streptococcus, then we would assume that it is round-shaped. In fact, it would be because it has that in the name. So coccus is spherical. We also then have a rod shape. And rod shape, so something that's quite long like this, like a rod, this is bacillus. We also then can have the combination of the spherical and the rod shape, which we would call coccobacillus. And that's kind of this not quite round, but not quite rod-shaped. It's more of an oval-shaped bacteria. Then we can get into details for more curved shapes. So for example, Vibrio. So Vibrio is a comma shape. So comma shaped, so what we would see is it kind of tapers off. So we have one longer end and then it kind of tapers off just like a comma. So our Vibrio is more comma shaped, and sometimes this isn't quite like a pointy area. It's rounded on both ends, but it's just much smaller on one end than it is on the other end. So our Vibrio, sorry, I'm kind of messing up my organization here. So we have coccus, bacillus, coccobacillus, and then Vibrio are the three different classifications there, shapes. Then we have our spirochetes. Or spirochete. And so our spirochetes are kind of what we think of as an old school telephone cord, where they actually very much spiral on itself. So our kind of telephone cord, it's very, very spiraled. And of course this would be, three-dimensional here. All right, so that's our spirochete, which is a spiral shape. And then lastly, we have spirillum. And spirillum is kind of a wavy shape. So rather than actually spiraling on itself, it's just kind of this squiggly shaped bacterium. So wavy or squiggle, if squiggle makes more sense, something like that, because it starts with an S. And then that's different between a spirochete, which is a spiral, and spirillum, which sounds very similar, but maybe we can remember that's a squiggle. It's a wavy line. So those are some of the different common shapes. Now I do want to speak just a moment to different arrangements. And we'll talk about this more in the next chapter, the chapter after that, in chapter 3, I believe it is, when we talk about different arrangements of bacteria. But I want to mention it now because we're talking about the common shapes. And I already mentioned, for example, Streptococcus mutans, just as a general example. Because when we're talking about coccus shapes and sometimes rod-shaped, we can then utilize different terms to identify the arrangement as well. So, for example, we can say staff. If we see staff at the beginning, then that's going to indicate that we find that in clusters. So if we say Staphylococcus, then that means that we are finding clusters of round-shaped or spherical bacteria. So Staphylococcus are clusters of round shape, so they're kind of grape-like clusters. On the other hand, as I've already mentioned, is strep. So strep is in chains. So if we hear Streptococcus, then what we're seeing are chains of circles. So if I draw that out, our Staphylococcus would be lots of circles kind of clustered together, and then some other ones clustered together. So this would be our Staphylococcus. And then over here, if we're talking about our chains, we really would see kind of long lines here. This would be Streptococcus. So we're seeing Streptococcus here, and then over here we're seeing Staphylococcus. So Staphylococcus would be our clusters of round-shaped. Sometimes we do see Streptobacillus or Staphylobacillus, not so much. We do see Streptobacillus, but then we also can see Diplococcus. And Diplococcus, di meaning two. So we can see kind of two spherical ones stuck to each other like that. And there's also quad and things like that, but we're going to hold off here at the moment and just separate between staph, strep, and diplo. So our staph is in clusters, strep is in chains, and then diplo is di, meaning two, so Diplococcus. And oftentimes, as you can see here in my example, we're talking about coccus when we're using these arrangements, and sometimes bacillus. We don't really see anything like Streptovibrio, that's not a thing. It's more Streptococcus, Staphylococcus, Diplococcus, and sometimes Streptobacillus. All right, so that is our bacteria section. So our kind of general overview on bacteria. Of course, throughout the semester, we're going to get in much more detail about that. So let's kind of back up and remember where we're at here. We're kind of talking about the different classifications, the different specifics of these different classifications. So we started out with our major group, our prokaryotic microorganisms, which means that they have no membrane-bound nucleus and they are unicellular. We separated our prokaryotic microorganisms into bacteria and archaea. We just spoke about bacteria, and now we're going to speak a bit about archaea. So if we talk about archaea, remember, again, since it is prokaryotic, it does not have a nucleus, so no membrane-bound nucleus. Archaea are also unicellular, which we already know because it's a prokaryotic organism. In this case, however, when we're talking about our archaea, our cell walls are not made of peptidoglycan. Our cell walls are made of pseudopeptidoglycan. Pseudo means fake, and then peptidoglycan. So recall that we had a group that were bacteria, and then upon further investigation, we separated out the archaea bacteria, which is now called archaea. When that happened, they noted that in the cell walls, remember that the structure was different. So that was one major difference between bacteria and archaea bacteria, which is now archaea. And so then what they called the cellular walls, what they're made up of, the kind of structure of cell walls, they called it pseudopeptidoglycan, meaning fake peptidoglycan. It's not quite like it, it's different now. And again, we'll talk about those differences. Like bacteria, however, they're found nearly everywhere. However, unlike bacteria, they are not pathogenic. So we so far have not found a pathogenic archaea. They are also different from bacteria in their genetics. So I'm going to say different from bacteria in their genetics. So we see major differences in their DNA and RNA that, again, we'll talk about in more detail. So in their genetics, in their metabolic pathways. So what they metabolize, so their different sources of energy, and we'll talk about that. And then also in their cellular composition. So for example, the pseudopeptidoglycan versus the peptidoglycan. And that's really all we're going to talk about Archaea at the moment. So again, no membrane-bound nucleus, just like all prokaryotic organisms, unicellular. Cell walls are made of pseudo-peptidoglycan, unlike bacteria, which are made of peptidoglycan. They are nearly everywhere, just like bacteria. However, they are not pathogenic, and they are different in other ways. So moving back up to seeing our prokaryotic microorganisms classification, now what we're going to do is we're going to move over and we are going to talk about our eukaryotic organisms. Microorganisms. All right, so eukaryotic microorganisms. Recall that eukaryotic means that there is a membrane-bound nucleus. And in the case of eukaryotic microorganisms, they can be unicellular or multicellular. So now what we're going to do is we're going to separate our eukaryotic microorganisms into further classifications, into smaller groups. So the first group that we are going to talk about are the protists. So the protists, they are kind of a catch-all group. So our protists are unicellular, and they are unicellular eukaryotes. Eukaryotes, which of course they are because they're part of our eukaryotic microorganisms, but they are unicellular eukaryotes that are not in the other groups, basically. So fungi, plant, or animal. And what we mean by that is, again, they're kind of a catch-all group. So if they don't fit nicely in another category underneath eukaryotic microorganisms, we put them in the protist group. And then what we're further doing, scientists in general, are breaking down the protist group into further classifications and smaller groups. So some of those smaller groups that we can talk about here, one that we're going to talk about, if we take our protists and separate it out further, we can separate it out into algae. And so algae can be either unicellular or multicellular. Algae are photosynthetic. So if you think about algae that you know, things that are growing in water areas, so in pools or lakes or ponds, they're photosynthetic. We can see algae coating things. They need to be exposed to sunlight in order to grow. Algae have cell walls, and their cell walls are made of cellulose. So cell walls made of cellulose. Now, cellulose is a particular specific type of carbohydrate. So we see cellulose in plant cell walls as well. So in plant cell walls, and we see them in algae as well. So algae can produce very useful things. So for example, carrageenan is from algae. You may see carrageenan in different food items that you've maybe looked at. It's used as a thickener, kind of a gelling type of thing. And then specific to us in microbiology, we get agar from algae. And this is a gel that we use in, whoops, not two L's, a gel that we use in agar plates, or petri dishes as they're sometimes commonly called. So algae can be quite useful in many ways, and it is considered a protist. It's a type of protist. Now, the other group of protists that we're going to talk about are protozoa. So we're going to zoom over here to protozoa. So protozoa, as is mentioned in the text, they are kind of the backbone of our food webs. Now, a lot of organisms are going to be utilizing protozoa for their original food source, and then they get bigger and bigger and bigger from there. Protozoa are very diverse, as kind of the protist group is in general. but protozoa are very diverse. Some of them can move. Many of them can move. And then the thing is that in protozoa, they can move in various different ways. So some of them move via cilia, which are hairs, basically, like little tiny hairs. So sometimes what we'll see with a protozoa is we'll see all of these little tiny hairs all around the protozoa, and then it allows it to move by beating those little hairs in a wave-like motion. It allows it to move through fluid. Some other ways that they can move are by using flagella. Flagella is like a tail. So, for example, we can have a protozoa that has a little tail on it that's going to allow it to move. Sometimes they have lots of little tails, lots of flagella on there that allow them to move in different directions. Or sometimes they can use pseudopods. Again, we see the word pseudo, and pseudo means false or fake, and then pods stands for feet. So false feet. And in this case, what happens is the organism, the protozoa, can kind of glob out a piece of its cellular material. It still stays within the cell itself, but it can take the jelly stuff in the inside, like the cytosol, and kind of push it out to make what looks like a false foot, like a little kind of arm there. So it'll be kind of like an individual cell, and then it'll kind of push some of its fluid in a direction to make it look like a little false foot. And then it'll maybe push another piece out. So we'll have false feet. And then what it can do is it can take this and it'll put all of its fluid in that area there, and then it allows it to move. So it'll filter it all into that area, and then it'll push out another false foot and allow it to move into that area. So it kind of looks like it's walking. So some of them can move. Some other characteristics of protozoa: some are photosynthetic, some are not. So some are photosynthetic, some are not. So some use organic compounds for their source. Some are parasitic. So if it's parasitic, that means that it has to have a host or uses a host to get its nutrients. Or some are free living, so they don't need one. They can live on their own and get all of their nutrients by themselves. And lastly, some are pathogenic. But most are not. So we have lots of different characteristics. Again, our protozoa are very, very diverse. So we can see that some move, some don't, some photosynthetic, some not, some are parasitic, some are not. But all of these are characteristics of protozoa, and then furthermore, they're all characteristics of certain types of protists, which, remember, are a type of eukaryotic microorganism. So another type of eukaryotic microorganism that we're going to talk about are fungi. So fungi are, excuse me, are eukaryotic. Again, that means it has a membrane-bound nucleus. In this case, some fungi are unicellular and some are multicellular. So unicellular. So a unicellular example would be like a yeast, for example. We have some unicellular like Saccharomyces cerevisiae is a unicellular yeast. Some are multicellular. A good example of that is what we think of when we think of fungi, which is a mushroom. That's a multicellular organism. So they look very similar to plants, but they're not actually plants. They're actually fungi. They're not photosynthetic. Plants are photosynthetic. They use the sun for their energy, for their carbon source, and then fungi are not. So mushrooms grow in the shade. So that's a characteristic. So not photosynthetic. Whether we're talking about a mushroom or we're talking about a yeast or any other type of fungi, they're not photosynthetic. And then the cell walls of fungi are made of chitin. So cell walls made of chitin. And chitin is another type of carbohydrate. So it's structured differently than our cellulose. Remember, cellulose is a structural carbohydrate we find in plant cell walls. But then we also see that in algal cell walls or in algae. In fungi, what we see is chitin. And chitin we also see is in the exoskeleton of insects, for example. So that hard exoskeleton in insects is what we find inside of the cell walls of our fungi. So then we can further break down fungi into different groups. So if we break down fungi, we're going to break it down into our yeasts, and we're going to break it down into our molds. And so just a little bit of their characteristics. Our yeasts are unicellular fungus or unicellular fungi. And so yeast can be both helpful and harmful, right? So our helpful, we can see we can use some of our yeast to make bread, or we can use it to make different beverages. So wine or beer or kombucha, all of those things are going to be utilizing yeast, also ginger beer, things like that. But then they can also be harmful, so they can cause infections like thrush, and they can contribute to things like food spoilage. Another type of fungi are the molds. Molds are typically multicellular. So these are multicellular fungus versus our yeast, which are unicellular fungus or fungi. Our molds have long filaments, and then these filaments form these visible colonies. So this is a situation where it is a microorganism, but we can end up starting to see these visible colonies. So we have long filaments that then form visible colonies, which you may be familiar with. So if you think of bread mold, for example, so you may not see the mold for some time, but those filaments are forming until they end up being that mold spot, that fuzzy mold spot that you end up seeing. So that's when we actually see those visible colonies, but they start out as a whole bunch of little filaments that you may not see at the beginning, but then they turn into that mold spot. And so then similar to yeasts, some of our molds can be helpful and some can be harmful. So for example, some that are helpful is we get a lot of our antibiotics from molds. So for example, penicillin and the other penicillins, amoxicillin, things like that, cyclosporin. Some can be harmful. So some examples of harmful ones is they can cause allergies. They can contribute to food spoilage, just like we talked about a moment ago. Food spoilage, all that doesn't make sense, does it? There we go. Food spoilage, like bread molds that make it so that it spoils and we have to throw it away. But then more concerningly, we can have this is related to molds, mycotoxins, and these can cause some very serious infections and disease. And we'll talk more about those in detail as well. So those can cause some serious problems and hospitalize people. All right, so those are the two categories of fungi. So here we'll just kind of zoom out again before we move into our next group. We've taken eukaryotic microorganisms, separated into protists and fungi, and then separated those further. We're going to then now separate our eukaryotic microorganisms into their last microorganism group. So again, remember we're talking about microorganisms here. So that means that we're not including animals, like big animals like us, humans or tigers or something, or bears. But we're also not including plants, some of the larger plants. So we're talking about microorganisms, so we're talking about our tiny stuff. So our last group of eukaryotic microorganisms that we are going to talk about are the helminths. And the helminths are multicellular parasitic worms. So note that it is automatically multicellular, is automatically parasitic. So parasitic meaning that it has to rely on a host to get its nutrients and to live. And they are the worms. And we had already mentioned that even though we're talking about microbiology here, we classify helminths in this area of microbiology because the egg and larval stages are microscopic. So the egg and the larva stages are microscopic. Those parts of their life cycle are so small that they are then classified as microorganisms. And so we talk about them in microbiology. So some examples of those, again, they're parasitic. So we see them, they can infect other animals like ourselves. So an example that's given in the text was the guinea worm. So someone can get that by drinking infected water. So drink water with the larva in it. But some other examples that you may be more familiar with are different tapeworms, for example. So there's a beef tapeworm, there's the pork tapeworm. You can get those by eating raw or undercooked meat. But then also something like pinworms that can be transferred just through physical contact. All right, so these are our eukaryotic microorganisms that we'll be talking about throughout the semester. So we can separate our eukaryotic microorganisms into protists, fungi, and helminths. We can further separate our protists into algae, protozoa, and we can further separate our fungi into yeasts and molds. So if we take a look at our two characteristics or two kind of areas here that we're talking about, our prokaryotic, our eukaryotic, and then you can see how we've separated those further. So these are all classifications that you need to be aware of, and these different specific characteristics for each of these different things. So one thing that we did not include in here are viruses. So where do we see viruses fit? Where do you think they fit? Do you think they're a prokaryotic microorganism? Or do you think they're a eukaryotic microorganism? Over here or over there. So here's where we're going to put it. We're going to put viruses right down here. So you can see where that is. You can see that it is completely separate. It is not a prokaryotic group or a eukaryotic group. Viruses are completely separate on their own, and that's because they are acellular. So A means without. So they are not made of cells. When we talk about living organisms, a living organism, one of the characteristics of a living organism is that it must be composed of cells. Viruses, therefore, since they are not composed of cells, are not considered living organisms. However, we need to study viruses, and they need to be put somewhere. And since they are very, very small, we cannot see a virus without the use of a microscope. They fit here in microbiology. So we talk in detail about viruses here in microbiology. So again, they are acellular, which means they are not made of cells. And since they are not made of cells, they cannot be either prokaryotic or eukaryotic, because both of those are classifications of living organisms that either have membrane-bound nuclei or do not. So viruses are acellular. Their structure, then, they're made of proteins and genetic material. Now that genetic material can be either DNA or RNA. And it's never both DNA and RNA. It's only DNA or RNA, and we'll talk about these in more detail. So viruses are made up of proteins and genetic material, and that's it. A virus, again, since it's not alive, it's not living, it's not made up of cells, it has to have a host. It absolutely has to have something else around it, something that it's living in order for it to be not living, but in order for it to continue existing. So it is inert, outside of a host. So another way of saying this, and we're going to get into some more details of this, is saying that it is an obligate intracellular organism. Obligate means it's obligated to be, meaning it has to, right? It's obligated, it has to. And then intra means that it's inside of, cellular, inside of a cell organism. So it's an organism that has to, is obligated to live inside of a cell. So in order for it to exist, in order for it to continue living, I put that in quotes, it has to be inside of another organism, inside of a host. And that is because it takes over the host's machinery. So it takes over the host's cellular machinery in order to multiply. So it has to take over the host cellular machinery because it doesn't come with its own machinery. It comes with proteins, it comes with genetic material, but it doesn't come with its own machinery. So it doesn't have any ribosomes, it doesn't have a Golgi apparatus, it doesn't have an ER, smooth or rough. It doesn't have any of those things that are within a cell that allows a cell to function. So it has to go into a host cell, it hijacks everything, and then it uses the host cell in order to make copies of itself to continue proliferating. And then many viruses are pathogenic. Some viruses are not pathogenic, but many are pathogenic. So some examples are the common cold, HIV is a virus, hepatitis, and Ebola. Oops. So those are just some examples of some viruses. All right, so that concludes our organisms, our classifications, our different types of organisms. So again, we have our prokaryotic organisms, our eukaryotic organisms, microorganisms, and then viruses. Because again, viruses are separate because they're not living, they're acellular. We have our bacteria and archaea, our prokaryotic, our protists, fungi, and helminths are eukaryotic, and then we can separate protists into algae and protozoa, and fungi into yeasts and molds. So you should know all of these different things, the different classifications. You should know the different things that are underneath each of these classifications and their specifics. The last part of chapter 1 that we are going to talk about are just some of the different studies and the terminology for the different studies in microbiology. So first of all, when we say microbiology, right, we're in microbiology, is the study of microorganisms. We said that at the beginning. So again, micro meaning small, so it must be seen with a microscope, and biology. Biology is the study of life. So the study of living small organisms. So the study of microorganisms is microbiology. Within microbiology, we have different studies. So we have bacteriology, and bacteriology, then, of course, would be the study of bacteria. We have mycology. Mycology can get a little tricky. However, if you remember when we talked about fungi, they can produce mycotoxins. And when we say different names for different fungi, we have mycobacterium. They can produce those are different types, but they are myco. So myco is related to fungi. So the study of fungi. Take away that mycobacterium part. I take that back. That's Mycobacterium tuberculosis, which is different. But, all right, so the next one, protozoology. So you can probably guess what this is related to, the study of protozoa. Yep. We have then parasitology. Which is the study of, you can probably guess this somewhat, parasites. However, remember what we're talking about here, helminths and other parasites. Then we have virology, study of, what's your guess? Viruses. And then lastly, immunology, the study of the immune system. So ology, perhaps you've guessed now, ology is the study of, right? So that's why we can separate microbiology. So ology is study of, bio is related to living things, and micro, so small living things, the study of. And we can separate these things as well. Bacteriology, so obviously bacteria, and then ology is the study of bacteria. So anything with ology there is going to be the study of, and then we can look at the first portion of it and see what it's related to. So immunology is the immune system, virology, viruses, etc. And so these are all of the different studies that we're going to be doing throughout the semester. So we'll be studying bacteria very closely, we'll be studying fungi, protozoa, helminths and other parasites, viruses, and the immune system. So you should be familiar with those different types of terms here and what they're related to. So this wraps up chapter 1. So remember that microorganisms are very small. Typically, in order to see it at the visible eye, we're looking at something like 100 micrometers. Most of them just need to be seen with the microscope. We can separate it into all of these different categories that we've gone through multiple times now, you should be familiar with. And then lastly, we're talking about the different studies here, the bacteriology, mycology, etc. So that's the end of chapter 1, and in chapter 2, we're going to move into different microscopes.